So it's my pleasure to introduce our next session, which is going to be integrating therapeutic advances and stakeholder perspectives into NSCLC clinical pathways. It'll be presented by, discussions and implementation. So Dr. David Jackman will be joining us on stage, and I did want to let everyone know that this educational activity is supported by an educational grant from Genentech and Novartis Pharmaceuticals. So thank you very much for joining us, and look forward to your presentation.
Thanks so much for having me. It's my honor and responsibility to talk with you a little bit about non-small cell lung cancer today. You know, I view these things as not an opportunity to kind of go through every single aspect of every decision we make as we try to think of a lung cancer pathway, but let's talk about a few key decisions of new information in lung cancer and how we think through these things. What data comes to the fore? How do we think through this so that we can help our patients as we develop pathways? This is me. I'm the medical director of our clinical pathways program at Dana-Farber. We've built our own clinical pathways at our institution with the help of VIA Oncology, who we use as our platform vendor. These are my disclosures for now. And these are our objectives. As we go through this, I don't know that I want to bog down. It's a very wordy slide. But basically, we want to look into some key decisions within non-small cell lung cancer and talk about the thought process there. And so I want to start by talking about, as we come to the table to make clinical pathways decisions, some things that we need to think about. And so I want to talk about stakes and stakeholders. So what's at stake? You know, so as we build pathways, and this is kind of old hat at this point, we need to challenge ourselves to think about what's the best care we can provide. And as we think about that, you know, best care for whom? You know, I think we think about pathways as helping with population management. What's the best care for the many? But also want to think about how pathways can help the person in front of me in the clinic. And so for that individual, are our pathways granular enough that they capture what's really in this person's best interest? We want to think about how pathways can guide our present, but also allow flexibility for the future. We think about that at the patient level. That is, if this is our on-pathway decision, what doors does it potentially open or close down the road for this patient? Will it make them potentially eligible or ineligible for future trials? And then also, what's the flexibility for us as a population? If we blow so much money by putting a certain agent in first line, are we potentially limiting ourselves down the road from a cost perspective? And certainly we want to think about efficacy, toxicity, and cost, and generally in that order. I think pathways should be comprehensive. They should be granular. We talk about trying to reduce unwarranted variation. We also need to think about pathways as allowing for necessary and medically or otherwise useful variation. And they need to be dynamic, living, breathing documents because medicine changes dramatically and quickly. And I think that through all of this, at least in our program, we want to avoid thinking of being off pathway as wrong. We want our providers to have the freedom to take care of the patient in front of them and recognize that not everybody fits within the square pegs of a pathway and need to feel free to veer off pathway when it's appropriate. Who are the stakeholders here? You know, as we think about pathways, you know, who does this affect? It certainly affects our patients more than anyone. It has impact for providers and their workflow and the care they provide. Impact on our institutions or our practices, and on payers and on industry, on pharma. And so as we think about all of these stakeholders, I think there's a certain level of transparency and consistency that would benefit everyone in the process. You know, so I think we need to think about and be open about who actually gets to the table and what information gets to the table. So capturing who's involved and what potential conflicts do they have. How does an agenda get set? You know, for industry too. You know, to feel that when a new label comes around or a new FDA approval comes around or an important phase 3 trial comes around, that data is going to be discussed at the next meeting. And so how do we create benchmarks for setting an agenda? What data gets considered, you know, and how do we decide upon that? Making the meal. You know, when we're actually in the process, what's the level of evidence that requires? Should it only be phase 3 randomized trials? Can lesser weight of evidence still be important? What tools do we use to consider value? And how do we quantify the inherently unquantifiable? You know, it's easy to look at numbers when you're talking about response rate or survival or cost. How do you put a number on the importance of losing one's hair or significant nausea? And how do you factor that in? And so these are important, and it's obviously going to depend on one's perspective. And it's going to be a lot more important to a patient or a patient advocate. And so how do we bring that into the conversation? And then who gets to eat? You know, where do these pathways go? And so how do we disseminate these decisions? You know, after we make these, it's important that we have a tool that allows us to educate everyone using the pathways. And who's the intended target, right? So when we think about our pathways, are they for academic institutions? Are they for the community? Are they for everybody around? And can we make them broad enough and, as I mentioned, granular enough to be useful to all of those different kinds of institutions? And so, predicting that we were going to have funny music to go with the questions, I was trying to think, you know, what's the right song to go with all the changes that have come in first-line non-small cell over the last year. And so I was thinking about the Byrds and was thinking about David Bowie and changes, and ultimately settled on God Only Knows. Because, I mean, I think there's so much going on right now, and I think we're still trying to get a handle on a lot of it. And so with that, I want to talk about where we are in non-small cell lung cancer. Where we were, now about 15 years ago, was platinum doublets. Throw it at the wall and see what sticks. Didn't work in everybody. Response rates of about 20 to 25%, and overall median survivals of about 7 to 8 months. They were all pretty much the same in terms of efficacy or lack of efficacy, depending on one's perspective, but they differed in schedule and side effects. And so that's where we were in 2002. No carve-out for biomarkers, no carve-out even for different histologies. And so where do we stand now? We think now about histology, squamous and non-squamous, and increasingly appreciating the difference in biology there. Within non-squamous, you know, we're asking, is there a targetable genomic change or not? With squamous, we haven't had the same success with regards to genomic changes, and so we're still mostly thinking about platinum doublets. Within targetable changes, within EGFR, we want to think about erlotinib, gefitinib, and afatinib all as having first-line indications, and how do we decide amongst those? Within ALK, crizotinib, and now ceritinib all have first-line indications, and new data is increasingly pointing towards a role for alectinib here. Within ROS1 and BRAF, these also have FDA labels for crizotinib in ROS1 and for a combination of dabrafenib and trametinib in BRAF coming over the last year. In those patients who do not have targetable changes in non-squamous lung cancers, we still think about platinum doublets alone or potentially with bevacizumab. And in both non-squamous and squamous, we think about, for patients whose PD-L1 expression testing is greater than 50%, we think about first-line pembrolizumab. A few other things there. In squamous and non-squamous, we can think about the addition of necitumumab. Officially, it's FDA approved, though it's not something that has gained a whole lot of traction. And then one last thing to talk about is there a role for pembrolizumab in combination with chemotherapy in the first-line setting for non-squamous tumors in a PD-L1 expression agnostic way? That is, doesn't matter what your testing was. We want to do this for everybody. And so is that ready for prime time? And so these are the things I want to talk about today. I want to talk about within ALK, want to talk about all the different agents and where we stand right now. Are we still sticking with crizotinib, the first-generation inhibitor, or should we be moving towards a later-generation inhibitor? I want to talk about the potential role of immunotherapy alone in those patients who are greater than 50% expressors, or potentially in combination with chemotherapy. And want to talk about platinum doublets and squamous cell carcinoma, and is there any variation within that to help guide the choice of medication? And so this is the section where we say, what were you thinking? Pembrolizumab in first-line non-small cell for those patients who are PD-L1 expression greater than 50%. So how do we think about this? So we go ultimately to the Keynote 24 study, in which patients with any sort of non-small cell lung cancer, squamous or non-squamous, were randomized, and those who specifically had PD-L1 expression testing that was greater than 50% with a specific assay, with the 22C3 assay, were randomized to receive either platinum doublet chemotherapy, the standard, versus pembrolizumab, a PD-1 inhibitor, and were followed till progression or toxicity. And what we saw was that in this particular set of patients, that response rate was favored with the pembrolizumab group, and that, contrary to some people's thoughts, the time to response was just the same. Some people think, immunotherapy takes longer to kick in, but that's not what we saw here. The median time to response was similar. The duration of response for the pembrolizumab group still has not yet been reached, at least at the time of this publication. And when we look at progression-free survival, it was significantly superior for the pembrolizumab group. When we look at progression-free survival on this forest plot, what do we see? We see that across most of the different subsets, it favored use of pembrolizumab. These few that are more questionable were female sex, current smokers, and most prominently, never smokers. The current smoker one was quite surprising because I think we think of current smokers, and usually heavy smokers, as being those more likely to respond to immunotherapy. And when we look at overall survival in the intent to treat population, that too favored use of pembrolizumab. With regards to toxicity, the immunotherapy, pembrolizumab, had fewer side effects, fewer serious side effects, fewer grade 3 and 4 toxicities. And so with all of that, as we make ourselves a checklist for this decision-making, right, we ask ourselves, do we have the information we need? In this case, we have a well-conducted randomized phase 3 trial for this population. Does it help the patient in the short term with respect to immediate clinical indicators like response and progression-free survival? And the answer is yes. Does it help the patient in the long term? Did it help with overall survival? And that answer was also yes, and that's despite crossover. Is it less toxic? And the answer there, too, was yes. Is it less expensive? That's a harder one, right? I mean, because I think it takes a longer-term total cost analysis and not just looking at the drug costs in this case. Because in those patients who get chemotherapy first, many of them are ultimately going to get immunotherapy down the line. And so are we necessarily saving money? I'm not sure about that. Does it compromise future care for these patients? And the answer there is no. It's not necessarily closing any doors down the line. If you get pembro first, chemotherapy will still be there for you down the road. And so with all of that, and given this, I think it's hard to argue against using pembrolizumab for this specific population of patients. Again, assuming that they're eligible for it, that they don't have autoimmune diseases. Next thing I want to talk about is, what were we thinking with respect to, is there a specific role for nab-Paclitaxel plus carboplatin as your doublet of choice in stage 4 squamous cell carcinoma? And so this is one that I think is harder to see, right? When we look at the data in the CONSORT trial that Mark Sosinski had conducted and published, patients were randomized to carboplatin plus nab-Paclitaxel, an albumin-bound paclitaxel, versus carboplatin with standard solvent-based paclitaxel. And what we saw overall was that the response rates the response rate was a little bit better in the nab-Paclitaxel group, but most of that, as we'll see in the next slide, comes in squamous cell patients specifically, but that there was not a statistically significant improvement in progression-free or overall survival. When we break down response rate, we see that the improvement was really in squamous cell patients and that there was very little difference in the non-squamous population. When we think about costs, so these are drug acquisition costs that I borrowed from the VIA online oncology cost calculator. And when we look at the difference between carboplatin and standard paclitaxel versus carboplatin with nab-Paclitaxel, there's obviously a significant cost difference over 6 months. And so here too, do we have the information we need? Was this a well-conducted, randomized trial? The answer is yes, with sufficient people in each arm. Does it help the people in the short term? The answer is, well, kind of, maybe. If you're a squamous cell patient and response is really important for you clinically, maybe you have a large mass that's pressing and causing shortness of breath, so their response might matter. In someone who's not symptomatic, you could argue response doesn't necessarily matter as much. If it doesn't improve progression-free, and as we see in the next line, does it help in overall survival? If the answer is there no there, well, then I think you could be justified using the standard Paclitaxel. Is it less toxic? Well, it's less toxic in certain specific things. That we know that nab-Paclitaxel is easier with neuropathy. It's easier in that it doesn't require steroids. So in certain specific toxicities, it was a bit less toxic. But in other things, particularly in hematologic toxicity, it was tougher. The schedule is also arguably tougher because it requires weekly visits instead of once every 3 weeks. Is it less expensive than nab-Paclitaxel? Clearly not. Does it compromise future care for the patients that get it? No, not necessarily. And so with that, I think you look at this and say, well, maybe I could justify it for our squamous cell patients who really need a clinical response. But in the others, I think we're hard-pressed to justify the use of this regimen. Next situation I want to talk about is first-line therapy in ALK-rearranged non-small cell lung cancer. And so ALK rearrangements were first described by Dr. Soda in Japan, and shortly thereafter, once these were described, Pfizer looks and sees, wait, we've got an ALK inhibitor on the shelf. It was actually a MET inhibitor, but it had known ALK activity, so let's give it a shot. And so Eunice Kwok from the MGH in Boston publishes this phase 1 trial, and the results are spectacular, so spectacular that the FDA actually grants accelerated approval on the basis of a phase 1 trial, which at the time was just landmark and groundbreaking. So thereafter, we've developed several new ALK inhibitors, second-generation ALK inhibitors like ceritinib and alectinib, knowing that, well, crizotinib's great, but it wasn't even designed as an ALK inhibitor. We think we can do better. So are we doing better? So this was the J-ALX trial, J being Japan. This trial was conducted specifically in Japan. And when they looked at progression-free survival, the alectinib group in the red line here, clearly doing better than crizotinib. And this year at ASCO, this is still the J-ALX data. When we look at response rate, alectinib did better with regards to response rate, not surprisingly. It's also better tolerated. So there were fewer grade 3-4 toxicities, there were fewer dose interruptions, and fewer drug-related discontinuations. So it looks like it's more effective and less toxic. And we saw similar data this year at ASCO when they presented the first results of the ALX trial. Again, alectinib, in this case in blue, is superior with regards to progression-free survival compared to crizotinib. And again, looking at certain secondary endpoints, here they looked at the cumulative incidence of CNS progression. This is particularly important in ALK rearranged non-small cell lung cancers, which tend to have a greater incidence of brain metastases. It's also telling because we know that alectinib gets into the CSF and into the CNS better than crizotinib does. And so not surprisingly, there was a lower rate of CNS progression in patients receiving alectinib. With regards to overall survival, the data is not yet mature, but from what we have so far, there's not a significant difference in overall survival. One could say, oh, the tail looks like it might favor alectinib, but at least we can say it's not worse. I think one of the concerns going into this trial was, well, yes, maybe alectinib is better than crizotinib in the short term, but that's not really the question we want to answer. What we really want to answer is, the standard would be crizotinib first, followed by alectinib, versus going straight to alectinib. And so it's not enough to just think about progression-free survival. You want to think about the longer term and want to make sure that going to alectinib first isn't better than getting it sequentially. And here what we see is there hasn't been a compromise of survival. If anything, it's trending towards the alectinib monotherapy as first line. From a cost standpoint, again, from the VIA Oncology cost calculator, it doesn't look like there's a significant difference. Again, if anything, at least by the prices that were available at the time of this. Oh, thank you. Got it. In terms of alectinib, it looks like it might be even a little bit cheaper. So again, when we put our checklist together, do we have the information we need? Well, mostly. We've got 2 really well-conducted randomized trials. What are we waiting for? I want to wait on I'd really like to see the survival data. Does it help the patient in the short term? Clearly, yes. Okay, that's annoying. Sorry. Does it help the patient in the long term? So far, it doesn't look like there's an improvement in overall survival. But like I said, we're glad there's not a decrement. Is it less toxic? The answer is definitely yes. Is it less expensive? At least per the drug acquisition costs from the prior slide, the answer is yes. And is it compromising future care? And as far as we can tell, it does not. It's not causing long-term toxicities that compromise what you get later. It's not screwing up potential eligibility for specific trials of interest. It's not leading to other stranger mechanisms of resistance that might wind up ultimately causing patients to do worse in the future. So with all of that, we think strongly about moving alectinib into the first-line setting based now on the J-ALEX and the ALEX data. And the last situation I want to talk about is there a role for chemotherapy with pembrolizumab as first-line treatment in non-squamous, non-small cell lung cancer? And so this is based on the Keynote 21 study. It was a randomized phase 2, so I want to bring your attention specifically to the fact that there's about 123 patients, so 60 in one arm and 63 in another arm. So it's a small trial. Patients with non-squamous, non-small cell lung cancer of any PD-L1 expression, so they weren't enriched for a specific cutoff, were randomized to chemotherapy alone as a standard with carboplatin and pemetrexed versus chemotherapy plus the addition of pembrolizumab. And what did we see? Response rate was significantly better with the addition of pembrolizumab, and that's not entirely surprising. We know that they're all active agents, and we are not surprised to see that when we put them together synergistically, that they do better. For reference, recognize that pembrolizumab as a monotherapy in patients who were PD-L1 positive at 50% or greater from the prior study we said was 79%. And so, how much of this is really a reflection of synergy? How much of it is just what percent of the population was PD-L1 expressors, and we might have gotten this number anyway? I think that's harder to tease out. So we're glad that it's better, but I think we still need to understand this a little bit better. In terms of progression-free survival was better with the combination, but overall survival was not, and it doesn't even look like there's any significant change in the tail there. In terms of toxicity, well, here we see that, not surprisingly, 3 agents are more toxic than 2, at least in terms of some of the serious toxicities. The grade 3-4 toxicities were higher in the combination group. The serious toxicities were higher in the combination group, though it didn't really manifest as an increase in drug discontinuations or treatment-related deaths. And so this is, I think, a more challenging checklist. Do we have the information we need? I would argue no. This is a randomized phase 2. Although it got its FDA approval, which frankly I'm a little bit surprised by, because there's a phase 3 addressing this question that's already completed accrual, and we're just waiting for the data to mature. So I think it wouldn't have been a stretch to wait on the phase 3 trial. And so I don't know that we have the information we need, especially knowing that we're just waiting for the data from the phase 3 to mature. I would say let's see that data first. We've seen a lot of things in phase 2, especially in terms of novel combinations, look exciting but then didn't pan out. So let's make sure that we get what we think we're getting. Does it help the people in the short term in terms of response rate and progression-free survival? Clearly, yes. Does it help them in the long term? Does it improve overall survival? It doesn't look that way. Is it less toxic? It's certainly not less toxic. It's certainly not less expensive because we're throwing all the drugs together at once. Does it compromise future care? I put question marks here. What do I mean by that? I'm concerned that certainly from an academic standpoint, by moving into this so soon, are we potentially compromising our conduct of clinical trials, not just addressing this question, but other things? And then for an individual patient, if you get all the most active agents first, And if it doesn't look like it's improving overall survival, well, when you progress on carbo and pemetrexed and pembrolizumab, what's left? The answer is not a whole lot. So I want to talk a little bit now about what's next for pathways in general and lung cancer more specifically. I think we really need to sort through immunotherapy, and I think we're still very much wandering in the desert on this. We don't have a great biomarker. PD-L1 expression is okay, but not super. People looking at total mutation burden, I think there may be a role there. So what the heck are these? These are the four horses of Venice. And I put them there because they've been yanked around throughout history. They were first sculpted in the Roman Empire in the 1st or 2nd century AD. They were stolen and brought to Constantinople, and they were in the Hippodrome of Constantinople. They were stolen by the Venetians during the Crusades and brought to Venice. Napoleon stole them and brought them to Paris. And then when Napoleon was defeated at Waterloo, they were brought back to Venice. And that feels the way that companies and drugs are trying to claim the immunotherapy space in lung cancer. First it was, nivolumab, you don't have to do any tests, it's for everybody. And then it's pembrolizumab, look, in people with high expression, look, there's something biological here, let's do that. And I think we're ultimately going to move towards, is there a role for combinations, whether with chemo, whether with CTLA-4 antibodies? Is there going to be some role with thinking about this with radiation? Is there an abscopal effect? I think so many things we don't yet understand. And so I think we're going to get tugged around in the immunotherapy space for a while, and we're going to pretend that it's really important to make a pathways decision for this right now, but recognize that the pathway is probably going to change every 2 months. We recognize that, for the most part, we've been doing targeted testing based on what's approved. So once we showed that EGFR inhibitors were important, then payers are willing to pay for EGFR testing. And then, so it's kind of the tail wagging the dog. Once we get ALK and ROS approvals for crizotinib, then they're willing to pay for this testing, and now they're willing to pay for BRAF testing. And now that NTRK inhibitors are approved in patients with NTRK translocations, will they pay for this? As we think about, MSI high tumors, which we can see in small cell and maybe in non-small cell, should we be doing that? And ultimately, how do we move towards kind of more general testing and think about next-gen sequencing as a way to test all of these things at once, using less tissue, and as the cost comes down and the technology gets better, it may be not just more efficient, but even cheaper than doing all of these a la carte. But ultimately, I think the real future for pathways is data. I was talking to someone from Amazon Web Services a few months ago, and they said, in this day and age, if you're not a data company, you're not a company. And I think that's very much true. And I think as we think about healthcare going forward, I think it's really cute and easy to think about, look, EGFR mutation, we'll use an EGFR inhibitor. But I think we're barely scratching the surface of how we need to think about things. I think that we need to get and capture the entirety of genomic data, and it's going to tell us things that we couldn't previously comprehend. I think we need to tie all of our choices to outcomes so that it can then be fed forward to inform future decisions for future patients. And I think pathways can be an important part of that. I think that hopefully this can be the watering hole where all of these things come to drink. I think that decision-making genuinely is getting more complex, and we've got to appreciate that. That I think, we're only beginning to understand the interplay of different genes. It's not just that you have a KRAS mutation, but it's that you've got a P53 and an LKB, and how that interplay is important, how the stromal milieu can be important, how non-coding regions may actually play a role in things. And so I think we need to appreciate that our comprehension and decision-making ability is limited. And I think, that if anything, we're not thinking about enough things. As I tried to sit down and think about all the different variables that go through my head when I'm sitting in clinic and trying to make a first-line decision for my patients. Pathways aside, these are the things I want to think about. And then I called my cousin who, lives in his mother's basement and has too much time on his hands, and asked him, Mike, what do you think about when you're trying to figure out who you're going to start in your fantasy football team tomorrow? And when he told me the number of things he thinks about, like, holy crap, you know, and that's for something useless, right? And I think, you know, we've got to let data inform our decisions better, and we've got to find a way to capture this data and tie it into pathways in a meaningful way. And I was floored. I didn't care about the kicker. Why do you care about the kicker? Well, Dave, if the kicker's lousy, the coach is going to go for it more, and that might drive up his fantasy stats. Oh, well, that makes sense. And so with this, right, we think about, you know, that right now we're looking at patient and disease characteristics, some important landmark things, but not necessarily really detailed, right? We don't have pathways that are looking specifically at the potential interplay of sequence of therapies and what your creatinine hematocrit are at the given moment and you know, kind of higher level genomics. And then we want to tie this into treatment decisions, but ultimately, you know, want to tie this to outcomes. So we're not just capturing what decision is made, but we're finding a way to capture how the patient actually did, so we can amass big databases of well, patients with these characteristics have these decisions made, and this is what's happened, so what should we do going forward? And so in conclusion, you know, I think that pathways in non-small cell lung cancer continue to evolve as science and drug development advance. We have challenges that remain regarding making meaningful comparisons, especially around value. You know, we find it incredibly hard to think about value and tools, in part because even when we want to use our own numbers, it's hard to get total cost of care. We can't get stuff that happens outside our walls. And so when our patients visit other hospitals or emergency rooms, we miss that. You know, oral therapies are hard to capture, you know, and because they're getting them filled outside, not within our walls. And that pathways really should play an integral part in guiding future care with respect to capturing, disseminating, and understanding data better. And so with that, these are my acknowledgements. This is my pathways team. These are the pharmacists who help with our build. These are all the doctors who are involved in building Dana-Farber pathways. DFCI leadership support is a euphemism of people I answer to. And this is via our friends at V Oncology, especially Kathy Loki, Ed Rogers, Stephanie Simon. That's what we got. Questions?
Thank you, David. So please make your way to the microphone if you have questions.
I'm just curious how, since we are talking mostly about stage 4 and stage 3, nothing has changed in stage 3 for decades. How are the results of the Pacific trial presented yesterday at ESMO are going to impact on your institution's approach to stage 3?
I think that's a super question. I think that we have our lung meeting specifically scheduled this Thursday in part to address that. So I don't want pathways to reflect just my decision making. I want them to reflect our group. So I don't know that I can tell you as yet what our institution is going to do with that. For those of you who don't know, this is regarding darvalimab. So it's another immunotherapy agent being used in stage 3 disease that was unresectable after chemorads as, quote, a maintenance therapy. To be frank, I've seen some of the data, but not all of it. So I don't know that I have a specific answer for you yet, but we will. All right.
So while I'm going to maybe indulge for a minute. You were alluding to toward the end of your presentations a little bit about how you see the future of pathways and that triangulation between treatment decision, patient preferences, outcomes. Could you maybe elaborate a little bit more on what that future looks like? How do you envision those factors being tied together in an integrated way to incorporate pathways into clinical workflow, but also help them use real-world evidence or other outcomes data to sort of inform the pathways and ultimately, I guess, inform the decision support and the shared decision-making environment?
I think it's a real jump to think that we're going to get to a point where we're capturing so much data that we are, as physicians, going to be comfortable with real-world data in some black box telling us what to do without any input into it. What I foresee is something, one step before that. Being able to capture real-world data and then feed it back to pathways committees and in a kind of more grandiose way saying, when we have this specific type of patient with this specific genomic signature, it looks like there may be a signal for this kind of regimen. And so do we need to alter our pathways to consider that? So there's still an expert human element being involved in it, and that pathways can be the mechanism for discovery, but that it's not being done in a black box kind of way.
Thank you. Question?
Sure. So since one of your questions was whether the treatment would in any way affect future treatment, so let's just look at the ALK inhibitors. Would you consider a certain sequencing in the use of the ALK inhibitors? Because, as you know, we're going to probably have 8 to 10 or 12 of these in a short time.
Yep. So I think that's an incredibly important question. I think a lot of that is going to come from our understanding of the mechanisms of resistance and what happens from one to another. We know that with crizotinib, there are a multitude of mechanisms of resistance, though many of them are secondary mutations in ALK, to which things like ceritinib and brigatinib and alectinib can hit. And I think what we need to better understand is after that, if you're on alectinib or you're on brigatinib or what have you, what's the mechanism from that? And will lorlatinib then be able to hit that, if and when it comes to the fore? And so I think that this is going to be incredibly important. I mean, it's going to be defined by science and not by, just cost considerations. That I think we're going to need to understand what the optimal sequence is going to be. And I think it's also important for us not to think that we can extrapolate from one set of targets to another. That is, we can't say, oh, well, we're bringing a second-generation inhibitor into the first-line setting in ALK. Let's do that in EGFR too. No, I think the mechanisms of resistance are incredibly different there, and so we need to think about that completely separately scientifically.
If I may ask you a question about scale, when you think about developing lung cancer pathways at your institution at Dana-Farber in Boston, how realistically do you think those can be applied? So, for example, can your practitioners be on the same page as a solo rural practitioner in Maine, for example?
I think that's a great and fundamental question for pathways, and hopefully role for pathways. I think that we and many of the pathway developers, around the country are trying to develop pathways as a means to take the expertise from places like Dana-Farber or what have you, and bring that to clinics and solo practitioners throughout the country. So this is fully intended to be adapted everywhere in many ways, but we also need to recognize that there are some things that are best done at a major center. And so whether that's things like bone marrow transplant or whether that's something like Whipple for early-stage pancreatic cancer, we need to, when appropriate, be saying, don't try this at home. You need to send folks into a major center for this kind of thing. But for much of cancer care can be effectively delivered in the community, and hopefully pathways is a way to enable that. The other piece is I think it's important for pathways, as some of our prior speakers had mentioned, cannot simply be just, here's what the drug you should use in this particular setting. I think that pathways are an opportunity to bring in information about things you need to consider, about symptom management, about enabling doctors to not just choose the care, but deliver the care. And those are the things that I think can really be enabling and ennobling through pathways.
Thank you.